What would you know about maturation?
Thanks, Dale. I appreciate it. So yeah, I'm excited to present this. Unfortunately, my research didn't go exactly the way that I wanted it to, as research tends to go. And with the conference coming, I threw together what I could. But I found out some really cool stuff about the microbial communities in the composting process and how that changes over time. So one thing that I wanted to look at was not only the product of the biochar out of a composting system, but also the process, to kind of understand how exactly we arrive to the product that we will. And I was really inspired by a study by Cheng et al. In 2006. It got me started thinking about how biochar is affected by different metabolites of the microbes, and also the high temperature. Something that really stuck out from that study was that the potential CEC of a biochar mixed soil was increased to 90% by subjecting the biochar to a 4-month incubation period at 70 degrees Celsius. And so when I was thinking about how this applies to the compost, I'm going to jump back a slide now. That's biochar on the right side. That's biochar that's fresh out of the kiln. There's no biology, as one would suspect. And then after it's gone, I grew the one on the left in a green roof media with compost, and the biology is moving in. This is a confocal microscope picture showing fungi in blue, and then in the green is the bacteria. And so I was thinking, when we put biochar into the ground, a lot of focus in this conference has been about not just throwing biochar in the ground and hoping you get good results, but charging it. Doug Clayton's enlightened us a lot with his techniques, and there's been lots of good research with that. So I wanted to kind of see how this thermophilic biochar compost would affect its performance in the environment. And so I saw a lot of similarities between a thermophilic composting system and Cheng et al.'s study, namely of which was the temperature. That 70 degrees C that he incubated at 4 months. It's about the same time that it takes for the compost to mature at a very similar temperature. And something that I wanted to juxtapose that with was the soil environment, where you're not going to get those kind of conditions ever. And so how you can wait for biochar to mature in the soil, but what I wanted to see was how it would be able to distinguish itself from those soil matured biochars, because the conditions are so variable. With this extremely porous nature, what I wanted to compare, another thing in the study was inoculated biochar, where you take completed compost and mix it with biochar, versus biochar that actually goes through that thermophilic process because of nutrients going in with the water and the microbes doing their thing. So I wanted to look at it in three different vectors: the physical, chemical, and biological venues. Physical, I wanted to see surface area. Are those pyrogenous gases condensing on, and what does that look like after you get it out of the compost? And then something that I was really curious about that I'm working on right now is rate of water loss. Because as biochar comes out of the compost pile, it's going to have all the organic matter and humus substances all over the surface. So I want to see the rate of release over time. And so what I'm doing right now, and I don't have the complete data for this yet, so I didn't post it, but the technique that I'm doing is I have it in a growth chamber at 10% humidity at 90 degrees Fahrenheit. And I have it on a balance, so I can see the curve of water loss to see if that's going to be different between a composted biochar and a non-composted biochar. And then the chemical nature, that's pretty straightforward. And the biological one, this is the one that I'm really excited about, was looking at the microbial abundance as a function of time, as well as the diversity, and how that's going to affect the plant performance at different stages, as well as root growth, doing a split root plant study, as well as disease suppression. I'm partnering with Longwood Gardens, and they're doing some greenhouse studies using vinca and looking at Phytophthora in moist soil conditions with and without fertilizer. So they're working on that right now to see how that changes. So I used the Longwood Gardens composting facility, and I just wanted to end up with something where the compost was a 50-50 blend. So we just used some greens from their facility, wood chips locally sourced, and manure from down the street. And I wanted to compare it to perlite, because I was thinking, perlite doesn't necessarily, biochar is biological charcoal. So I wanted to find something that was of similar porosity in nature, similar kind of size, and see, what biochar has to offer in terms of CEC and absorption, all those other things that distinguish it. And this is where, I started learning a couple things about how to set up this process, where we found that the compost with the biochar has some issues with oxygen concentrations, where it was getting more anaerobic more quickly. And so that was different than the perlite. And then also temperature, the biochar wasn't getting as hot. So this is going to give me quite different results than I was expecting. And so in trying to figure out why we were getting the results that we were getting, we looked at particle size distribution. I think this had a lot to do with it, where our biochar was pretty it was much smaller than the perlite particles, and so I think that gave it less porosity in the compost pile. And you're going to have more water holding capacity in that too. So I wasn't sure that if that was affecting the temperature as well. So a bit of technical difficulties there. But we still got some cool data as far as microbial community abundance and diversities in the compost piles. And what we did for that was I took samples at every time that we turned the compost pile, and I extracted the fatty acids out and created fatty acid methyl esters, and ran it through a GC mass spec to look at characterizing the fatty acids to see what kind of communities we were dealing with. And so I'm going to go through a day 9, a day 14, and a day 26 sampling of each compost pile. So the perlite, I call it the VB, the variable biochar, where I have the thermophilic, versus the control biochar, which is CB, which is just the biochar mixed with completed compost right next to the compost piles. This is a PCA analysis, part of principal component analysis, and I really like this way to analyze the information, because what it allows you to do is it allows you to group and distinguish your species based on relative variance of their components. So if something groups together, then they're going to be very similar, and if they're more spread apart, then they're going to be more variable. And so what I found with this one was the most interesting trend to me was the fungi, as well as looking at the gram-positive concentration. So what we found was, let me see if the mouse works. Okay, is there a pointer?
The mouse does work. We can see it.
Can you?
Yeah.
Oh, I can't see it. That works. Ooh, there we go. Thank you. There were lots and lots of different fatty acids, and there were only a few that are indicative of certain kinds of microbes. So we looked at gram-positive, which was this triangle right here, gram-negative, fungi, and then all the unknowns were there. And so what we found was this trend where the fungi was going more towards the perlite and further away from the variable biochar. So I'm not going to go through all of these, but I just wanted to point out the trends that we found using that kind of analysis. Where we found in day 9 and 14, the variable biochar had a greater concentration of the gram-negative as well as the gram-positive, as opposed to the perlite. And then on day 26, we found that the distribution was more even, and there wasn't too much clustering toward one or the other. And with the fungi, they tended to go away from the variable biochar, more towards the perlite, more towards the control compost pile. And in day 26, it was more concentrated towards the compost. And so that was pretty curious to me as to why the fungi were behaving in such a way. And so we looked at the pHs of the different treatments, and something that we're looking at is the pH was much greater for the control biochar versus the variable biochar, and that might have to do with the way that it was set up, as well as the anoxic environment. The fungi may not have been too happy in that situation. So then we looked at, I just wanted to compare the biology that was happening on the control biochar, which was the inoculated biochar with the finished compost, versus the thermophilic. Yes?
Were the piles being turned during this process, or they were stacked?
They were being turned on these days, where there's the red diamonds right there.
That's going to affect those local communities, obviously, right? Carry them up.
Sure. And so that's why I did it over time,
right,
to see how that would be affected by that. I instruct the guys who were running the compost facility. I just told them, turn everything at the same time, because they like to do it by oxygen percentages.
Yeah.
But since the variable biochar pile was so different in that, there's some kind of variation going on there. And so this is something that everybody was suspecting. I just wanted to provide the numbers for it, where we looked at all the fatty acids and found that there was a huge difference between the variable biochar treatments. And so this is where I took the biochar particles out of the compost, separated it, and then looked at the communities on the actual particles themselves. So you're going to see a lot more life going on in the thermophilic biochar particles rather than the static inoculant. And that correlation became a little bit weaker on day 14, and then on day 26, it became a lot stronger. And so I think that had to do with just the maturation of the pile and things just moving in. And so something that I wanted to look at was the compost inoculation versus thermophilic composting. Obviously, a lot more going on in the thermophilic compost pile. And what I'm working on right now, I don't have the data for it, but I'm really excited about this one, is doing a split root study to understand the propensity of the roots to grow toward one medium versus the other. And I'm going to be using this program called Easy Rhizo that I'd just like to make known to the community, because I think it's a very powerful tool that we could use to do root architecture mapping, where I'm going to have a variable biochar over here versus a perlite compost, and I have all those juxtapositions to look at. Will the biochar create more root growth on this side versus a perlite char? And just start to understand that, especially with our knowledge of what kind of microbial communities that we're introducing in based on that fatty acid analysis. So I apologize for the lack of data, but this is what we could come up with, and thank you very much.
A quick one. Are you doing like plate counts on the microbes, or are you doing just a qualitative, not a quantitative?
That was a quantitative test with the fatty acids. With the greater concentrations, that's how it was pulled toward the different treatments. So when you saw the arrow, the longer the arrow, the greater difference between one treatment versus the other. So when the arrow is this long, there's a slight difference, whereas if the arrow is that long, when it's control versus variable, then it's a greater, it's vectors. So it's a greater pull towards that. And that was based on the intensity of the spike in the GC mass spec on that fatty acid.
So it wasn't really a quantitative on the organisms, though?
So for the gram-negative, they all share one specific kind of unique fatty acid.
Right, so you're looking at the acid.
Mm-hmm.
That makes sense.
Yeah.
So reading that slide right about the pH, that the compost pH was actually as high as 9 or 10?
Yeah, it was a more basic pH.
That seems very high for a compost. Would that be acceptable as a pH level for a compost that was sold for use in landscaping, or would this be? Yeah,
Longwood Gardens. That's the compost they use for their entire facility. So what I just said is, hey, do whatever you normally do, just throw in perlite and biochar. So I don't make that distinction. I let them do that. I wanted to see the more commercial application. They're a very powerful horticulture organization. So I figured just let them do what they do, and I'll just throw in my biochar.
Yeah, I might have missed the very beginning. What were the composts being made from?
Chips, green waste, manure, and biochar, and perlite.
Anyone else? All right, well, thanks.
Oh, one more from Kelly.
Can you characterize the biochar a little bit? Maybe I missed that.
Right, it's soil reef biochar. So I wanted to use something that was out there on the market to just go with the the commercializability of that. So they do pyrolysis at 550 for 5 minutes with Condorosa pine. All right, thank you.
All right, thanks a lot.